Okay. Pause the recording. Okay, everyone, welcome to the Digestive System lecture. Our agenda is to discuss the mechanical and chemical breakdown of food in order to ready those foodstuffs for absorption in the body. We are very much, I always had a friend that laughed at this, we're always, as humans, we are a tube within a tube. We look like a tube, and then from mouth to anus, we're a tube as well, right? And that tube inside of us is known as the alimentary canal. The alimentary canal goes from our mouth or our oral cavity to the pharynx, the back of our throat, down the esophagus, right, our food tube, down the stomach, into the small intestines, then the large intestines, then the rectum and anus, the anal canal. That is the tube within the tube. That is the alimentary canal, and the accessory organs that feed into it are the salivary glands, the gallbladder, the liver, and the pancreas, right? Those are the four glands that are not in the tube, but send little tubes with materials to digest the foodstuffs that go through. If you'll open up to page 442 of the textbook, we're on chapter 12, Nutrition and Digestion. I want to tell you that there is a nice unit we begin the chapter with on nutrition, eating disorders, proper nutrition, vitamins, minerals, how to have a diet that is healthy. You know, I just had my blood test, very good, I'm happy. All things are good. You got to keep track of your own body. You've got to be an advocate for your own health. The most fundamental way to stay healthy is to eat right. And from page 422 to 442, that's 20 pages of that chapter, I have that nutritional unit. And I just want to say that I'm not going to test on that unit, all right? Instead, what I'm going to do is, sorry, I had to put the cat in, wretched beast that he is. Instead, that section is going to be for you and you alone to look at. It begins with the case of the sweet breath date, where you can reflect on a person who has an eating disorder. And you can go through, and I would just thumb through it. The importance of water, the importance of the macromolecules. We've kind of done that in the chemistry section in A&P1. We look at the Neolithic diet, which is, you know, kind of the caveman diet, which our body for 90,000 years as humans has physiologically evolved to adapt to a world that is very different than the world we live in, where there's Twinkies, ho-hos, high fructose corn syrup. We talked about that in A&P1. Those of you who didn't have me for A&P1, you know, we discuss a bit of nutrition when we talk about the chemistry. So this would be a nice review for you guys, all right? Those of you especially who didn't have A&P1. But those who did have one, it is still a good application of our chemistry. So check that out. But what I'd like to do with this lecture is to just begin with page 442, the digestive system, how humans break down and absorb food. Right? And so we've gone through an overview of the human digestive system. Figure 12.14 gives you that overview of the alimentary canal and the accessory organs. All right, on page 443, check it out. And know the general structure of the digestive system, where things are in relation to each other. That first the esophagus comes, then the stomach. And if you look closely, we're going to, through the chapter, we're going to look at each organ in details. So let's talk starting with the mouth. Food enters the mouth, and again, look on figure 12.16. I repeat, figure 12.16 gives you an overview of the organ system, the digestive system, and also gives the accessory organs and what they produce in each area. So we're starting with the mouth, and if you look on figure 12.16, the salivary glands empty juices into the mouth. Salivary glands have an enzyme known as amylase. Amylase breaks down starch. When food enters your mouth, It actually triggers a nervous reflex, which causes those salivary glands to make saliva. And, you know, in class, usually I ask, how many people have heard of Pavlov's dog? How many people took psychology? Basically, in that experiment by Pavlov, a dog and a bell and a steak are associated. So the stimulus of a bell elicits salivary production in the dog as it's paired with a piece of steak. All right, so it's what we call a conditioned reflex. We all smell bacon or a steak or whatever it is you like, and then it triggers a nervous reflex to make salivary juice, right, in order to begin digestion. And so most of us are familiar with salivating. When you want something, that's what happens. It's a stimulus reflex. SR stands for stimulus. The stimulus, let's say, is a tasty steak that you're grilling up. You smell it. And the reflex is saliva is produced. In the Pavlov experiment, a bell is associated with the steak, and you kind of learn that when you ring a bell, boom, it means the steak is coming. So then you learn the response. It's a little bit of psychology, but getting back to just the physiology of all this, and there is a psychological component to food, desire for food, that is very strong in our bodies. In addition to that, there's a psychological component to an aversion for food. Anyone ever get food poisoned? You know, raise your hand. I did, Voorheesville, New York, 1999. Terrible. And my wife and I split the special of the day, and it was well, we both split food poisoning. You know, that was a bonding moment, but it was terrible. And I will never go near veal parmesan again. You know, it's disgusting now to me because I think it was Staph aureus, some sort of infection that way. But regardless, to make a long story short, we produce about over a liter of saliva any given day into our mouth. And the saliva contains three things: mucin, buffers, and salivary amylase. Let's go to the amylase first. That's the head honcho of this. The salivary amylase breaks down starch into simpler polysaccharides, all right? And the polysaccharides, if you put a cracker in your mouth and you hold it for 10, 15 minutes and let the saliva work on it, it'll start to taste a little sugary. The starch is broken down in the cracker into polysaccharides, which taste a little bit sweet, more simpler sugars. Let's say they are broken down, but not into the simplest form. So anyway, so in other words, what should you take home from this? The mouth begins carbohydrate digestion. And that's again in figure 12.16. It gives you that idea that chemical digestion begins in the mouth, but also mechanical digestion. Mechanical digestion means chop, chop, chop food into smaller pieces. Why into smaller pieces? Well, my grandmother always had teeth that would fall out when she was eating. And it always reminded me of, well, why have teeth? Basically, you take a big piece and make it smaller. What's the point? It increases the surface area. That's the point. The take-home message is mechanical digestion breaks food into smaller units so that the surface area is more exposed to the enzymes. And when those enzymes act on more surface area, they're more efficient. That's the point of mechanical digestion. Mechanical digestion occurs in the mouth, so does chemical through salivary amylase, mechanical through your teeth. And we're going to talk about the teeth at the very end as a specialty topic. Did my research on teeth and oral bacteria. Anyway, mucin is also found in the saliva. It's a slippery glycoprotein that lubricates the mouth. And finally, there are buffers in the mouth that neutralize acids. They take the acidity of the mouth. And I used to do a pee, poop, and spit lab when I taught micro, where we would look at how acidic a mouth is. And the more acidic your mouth, the more dental caries or cavities you're going to get, or you have a propensity to get. So more acidity means more breaking down of enamel. And my own research kind of looked at the different types of bacteria that and their responses to fluoride and how much acid production that they engaged in. That was my area of research. And I found one type was more dangerous than another. So the mouth buffers, all right, the saliva in the mouth buffers the natural acidity of the bacteria in there that they produce. Mouth has generally a pH of about 8. So pharynx, I don't have a lot to say about. That's the back of your throat. It brings air to the trachea and it brings food to the esophagus. On top of the trachea is something called the epiglottis. And we said, if this is my trachea, this little hole, the epiglottis is a flap, like my hand is a flap that covers that trachea when food goes down, you see? So the epiglottis is like this, patent or open at any one time. And then when food goes down, when you swallow, the epiglottis covers the trachea so that you don't swallow the food or liquid. Anyway, the food then travels into the esophagus. A fancy word for the esophagus is food tube, conducts food from pharynx to stomach. The top is voluntary striated muscle, and the bottom of it is involuntary smooth muscle, which is why you can drink, you can drink a soda upside down, because at first you have voluntary muscle, striated, right? You swallow, and then the rest of it just moves peristaltically. Peristalsis means the rhythmic contraction of muscles to propel food through the alimentary canal. That peristalsis starts at the bottom of the esophagus. I repeat, peristalsis is wave-like movement of food through the canal, and it begins at the bottom of the esophagus. That's where involuntary smooth muscle propels food through. And you can really digest and bring all your food in being upside down. I mean, that's why you can, in space, propel food through your body. There's no gravity. It's not sucking you down. So, interesting. Okay, the stomach. Let's go on to the stomach. Let's take a look at the stomach. On figure 12.20, the stomach's anatomy shows that it's an elastic sac. It's generally on your left side of your abdominal cavity. It is generally stretchable. What do I mean by stretchable? Stretchable means able to accommodate liquids up to 2 liters in amount. So that's pretty big. Stomach is acidic. The acidity kills bacteria and digests the glue in the meats and plants that you eat. It's very important to kill the bacteria. You have, remember we said in A&P1, if you take your finger, right, that's about 1 centimeter, your fingernail, any area on the surface of the earth on average has 100 million bacteria, 10 to the 8th bacteria per square centimeter. And I consider that a lot. And so if you actually become paranoid, you have to sterilize your hands at every one moment. So microbes are our enemy. I've always said that. I've always predicted that we would have trouble with the microbes. And I don't want to talk about corona, but we have trouble with the microbe, right? And I didn't even include viruses in what I said. I said 100 million bacteria per square centimeter. All of that is coming into your body. You're eating it. And so what happens? You've got to kill it. You've got to kill it with the gastric juices of the stomach. So a pH of 2 is very acidic, even maybe a little more acidic than lemon juice, So important. Let's take a look now at the structure of the gastric cells. If you take a look at figure 12.21, it shows a gastric pit, all right, a gastric pit. It's on page 450, and I show the histology as well of the gastric pit, both in figure 12.21 and 12.20. Take a look at 12.20, page 450. You can see there's mucous neck cells, there's parietal cells, and then there's chief cells at the very bottom of the gastric pit. The chief cells make pepsinogen. Pepsinogen is the enzyme that breaks down protein. P for protein, P for pepsinogen, right? And I just want to say that proteins are not digested anywhere until we hit the stomach. And so now parietal cells, right above these chief cells, make HCl, hydrochloric acid. So when pepsinogen is made, it travels up the gastric pit. Hydrochloric acid attaches to the pepsinogen and gets rid of the tail of the pepsinogen, turning it into something called just plain pepsin, P-E-P-S-I-N. Pepsin is the active form of the enzyme that breaks down protein. Pepsinogen cannot act, all right? Why not? Why not just make an enzyme right away that can act? If you made pepsinogen, it would break your own cells down and you'd fall apart. The stomach would be eaten up from the inside out. So it has to make the stomach has to make an inactive pepsinogen to be produced, sent out of the gastric pit. The parietal cells make HCl to activate the pepsinogen, turning it into pepsin, and then it goes into the stomach, breaks down protein. And all of this acidity has to have some mucus around it so you don't rip apart your stomach. And that of course, is mucous neck cells that produce, mucous neck cells produce bicarbonate-rich mucus that protects the lining of your stomach. Isn't that a beautiful system? You can really appreciate the stomach now that you've looked at it. Figure 12.20. Now, if you note in all these chapters, there's these little yellow text boxes to chat about different things. Does putting hot food in the refrigerator ruin the food? You know, and you should read that and think about it. There's pros and cons. If we take a further look, moving on from, before we leave the stomach, just so you know, if you look at the anatomy of the stomach on figure 12.20, there's a cardiac region on top and a pyloric region on the bottom. The cardiac region has a cardiac sphincter. Everyone go like this with your finger. You know, it's a little round muscle, a sphincter, and opens and closes to allow things in and out. Cardiac sphincter is on top and the pyloric sphincter is on the bottom of the stomach. When the cardiac sphincter is open, it allows a little bit of liquid through, and that gives you heartburn. GERD, gastroesophageal reflux disease, if it happens too much. And the pyloric sphincter, if that's open too much, can give you a duodenal ulcer. Cardiac sphincter can give you ulcers of the esophagus and damage the esophagus. We'll leave it at that. It can, in some cases, lead to something known as Barrett's esophagus, and 10% of those cases lead to esophageal cancer, which is a deadly cancer and you know, serious cancer. You know, the rate of survival, I think, is 5 or 10% after 5 years. So, you know, nothing to sneeze at. Now, this here is pepsinogen. This is a little review of what I just said. Pepsinogen is broken down, right, by HCl, hydrochloric acid. In other words, the OGEN part, the tail, polypeptide tail, over the active side of the pepsinogen is broken down and turned into pepsin, and that makes an active protein digester. Last but not least, something I didn't mention is the enteroendocrine cells of the stomach. You know from the word endocrine, it's got to make what? Hormones. Hormones are produced that regulate the stomach's activity, and gastrin, endorphins, cholecystokinin, which is abbreviated as CCK, and somatostatin are all the hormones that regulate the stomach's activity. Keep in mind, some of them, like CCK and somatostatin, will slow the stomach's activity and allow you to digest things more slowly. So when, like, for example, fat comes into the stomach and the small intestine, cholecystokinin is released and slows the peristalsis rate down. So, you know, there's regulation in how quickly your stomach digests. Another thing to note about the stomach is the intrinsic factor. Intrinsic factor is something, a chemical needed for intestinal absorption of vitamin B12. It's needed, and vitamin B12 is needed to make red blood cells. So the ability to form your red blood cells is dependent upon the intrinsic factor being present in the stomach for vitamin B12 to be absorbed. When the stomach has damage and B12 isn't absorbed, red blood cell production is inhibited. Okay, so that's intrinsic factor. If we continue on to figure 12.22, and 12.23, we look at the structure of the small intestines. Food leaves the stomach through the pyloric sphincter into what we call the duodenum, or the top part of the small intestine. And in the duodenum of the small intestine, the duodenum has your digestion. All right, the small intestine is actually narrow, but it is long. The large intestine is wide, but it is short. All right, and that's why we call it large, because it's wide. Small intestine is actually narrow. That's why we say it's small, but it's very long. It's elongated, 18 feet, roughly 18, 20 feet. And if you take a look, most of the digestion occurs there. Look at figure 12.25. If you take a look in the small intestine, carbohydrates, proteins, and lipids are all broken down by pancreatic enzymes, and they digest all of the macromolecules. Stomach is only protein, mouth is only carbohydrates, but the small intestines do it all. If you remember anything about enzymes, remember that. So let's talk about starch first. Pancreatic amylase is released, and if you look at the structure of the small intestine, you will see that it has little hills. The hills are known as villi. Villi increase the surface area of the intestines to allow more enzymes to be released and more enzymes to be absorbed. Let me go over the general structure. The duodenum here is the first part of the small intestines. That's where these enzymes are released, as I've said before. The jejunum and the ileum are the next two parts of the intestine, and that's where absorption takes place. These many folds of the small intestine are seen in figure 12.22. And at the bottom, you can see the villus is a hole with, is a hill with little hills on top of it. The hills are known as microvilli. So what we do is we increase absorption and secretion of enzymes tremendously by increasing the surface area using these folds, right? These folds increase the surface area tremendously. So, you know, there's a disease where a child is born without the folds. We call it a flat topography. Topography means landscape. A flat topography leads to death within, days, because you can't absorb. You must have those folds. Very important for absorption and for secretion. So again, I just got a little overexcited, got ahead of my enzymes, but I just wanted to show you that anatomy. All right, if you take a look at that anatomy. And if you also take a look at the anatomy of the intestines, you see that there are longitudinal and circular muscles in what we call the muscularis externa, the wall of the small intestines. Again, these muscles contract, causing peristalsis, or movement through the long, small intestines. Let's go over now the different macromolecules and who digests who. Okay, number one, starch is digested first in the mouth by salivary amylase, then in the small intestines by pancreatic amylase. All right, and starch is broken down into maltose. Then we have intestinal brush border enzymes. Intestinal enzymes such as maltase, lactase, sucrase, which break down these simpler disaccharides into monosaccharides. Maltase breaks down, what do you think? It ends in ASE, so that makes it an enzyme. Maltase breaks down maltose, sucrase breaks down sucrose, and lactase breaks down lactose. Anyone lactose intolerant? That means you don't have the enzyme as an adult to break down lactose, which is generally dairy or milk sugar. We are indeed the only animals that drink milk as adults. However, I want to say that the more your genes come from the east or the south of Europe, in other words, Asia or Africa, the more likely you are to have that lactose intolerance. I want to tell you that lactose intolerance, we have one person who's lactose intolerant. Not 70% of all lactose intolerance diagnoses are incorrect, a study showed. So I want you to really be careful about it. I know that you're drinking the milk and then you're getting the symptoms, but you don't know for sure. you have to really do like a diary, and, you know, you still drink milk and cheese because you want it. I mean, I love milk and cheese, but I'm saying that it may be something else. I had a friend who swore he had lactose intolerance. He actually had a bile duct that was clogged with sludge. I doubt at your age that you're clogged with sludge. So, you know, you got to be careful. You have to really keep a diary of what causes what. So I want to keep that clear. You know, as you eat, do you always get it? What do you get? And, you know, sometimes, for example, you're eating the dairy and it's the fat that your liver doesn't like. So it can be other things. It doesn't mean that you don't have it. You know, you may have it. And then they have those lactase pills. Do you take the lactase pills? You know, maybe. You know, they have some effectiveness. So that's interesting. I'm on a tangent, so let's march on. Proteins. We begin protein digestion with pepsin in the stomach, and we complete it in the small intestines. All right, what enzymes are involved? Trypsin and chymotrypsin break polypeptides, which are large proteins, into smaller polypeptides, which are small proteins. And the small proteins are broken down by aminopeptidases and carboxypeptidases into individual amino acids, which you know from chemistry is the individual single unit of the protein. Nucleic acids are broken down by nucleases made by the pancreas. So again, we see the pancreas playing the major role in its digestion of macromolecules in the small intestines. Last but not least, I have a very fun picture. I didn't love the picture on figure 12.23, and Kendall Hunt Publishing put that in. It's okay. It's just like each thing gets pulled in to each intestinal cell. Amino acids, fatty acids, simple sugars. But I would like them to put this picture in. But you have it in your notes for your reference. If you look on figure 12.24, I have a picture of a fat globule getting broken down side by side with a liver. The liver makes bile, okay? Liver makes bile. And bile salts, I love the picture. It shows how a fat globule pulls apart with bile that's attached to it. Bile has like hydrophobic and hydrophilic parts, which pull apart bile. Bile is broken down by, bile mechanically breaks down fat, but it doesn't chemically break down fat. If you look, it makes it into smaller droplets. And that increases the surface area of the fat, allowing pancreatic lipases to actually do the chemical breakdown of fats into fatty acids and glycerol. All right. So again, if you look at figure 12.24, that's a picture of the liver, and you can see there's a right and left lobe, and you can see there's a gallbladder. The gallbladder is always green. In real life, too, it has a green look. And if we did the pig dissection together, I'd show you that. So anyway, maybe we should bring the pig to our pizza party when we come back. I don't think that'll be appetizing. Maybe we better not. We'll just leave it to the lab, to the video. But anyway, make a long story short, that's your liver. The liver makes bile. The bile is stored in the gallbladder, and the bile is released into the duodenum of the small intestine, the top part of the small intestine, and breaks fat down into smaller parts mechanically. And then the pancreatic lipase comes in and breaks it down physically or chemically into fatty acids and glycerol. All right, last but not least, I want to end with the large intestines and take a look at figure 12.26. The large intestines are pretty large. They have this general upside down U shape. That's where you get a colonoscopy after age 50 or at age 50 to screen for polyps. And I have a little picture of that process, figure 12.28. There's a colonoscopy where they're getting rid of a little polyp. All right, whatever. We don't want to get gross on the Zoom. But to make a long story short, everyone, the large intestine is rather wide, and food travels slowly through it. 12 to 24 hours it takes to get through the large intestines. 90% of your water absorption occurs in the large intestines. So really, it's the place where water is pulled out of the large intestines. And I will say that your poop basically is mostly 95% fecal dry weight bacteria, all right? I did a lot of my research in grad school for my master's on coliform counts and E. coli, which is, basically indicates sewage, indicates contamination of waters. Actually, in our area here, Schoharie County, Otsego County, Delaware County. But anyway, it's interesting studies. I always liked some microbial research, but I will tell you that the large flora of bacteria have a benefit. Remember we said in immunology that it protects against other types of bacteria. It also provides vitamin K for our bodies. These bacteria produce vitamin K, which is important in clotting, blood clotting. See, everything's kind of related. I think you're seeing that as we go through all these systems. So we call it mutualism. Mutualism from high school bio, I know you learned this, is that both organisms benefit. Like we give a house to the bacteria called our intestines, and the bacteria produce vitamin K for us. So it gives us nutrients. What is gas? Flatulation. When you have gas, basically it's bacterial byproducts. And I always found this really weird. Early Earth's conditions, before there was oxygen, before there was even life, mirrors your intestines, which is gross. And that's the type of bacteria that love it there. Primitive type bacteria, very primitive. You can tell someone you're very primitive when they give out some gas. It has CH4, you can write next to that, methane. H2 is hydrogen gas. H2S gives the smell of the gas, hydrogen sulfide, and some water vapor. So primitive. I had an uncle that was very primitive. He laughed every time he had flatulence, giggle around. He was a primitive person, definitely. So he wouldn't even know what the heck the word primitive means. But let's move on. Let's give an overview now. We're done now with all of the different organ systems and accessory organs of the alimentary canal, of the digestive system. Now I want to talk a little bit about the layers of the alimentary canal and do some specialty topics on the teeth, and the hormones, a little bit on the mouth. Let's start with an overview of the alimentary canal. If you take a look, and probably my best picture here is figure 12.22. There are clearly different layers of the alimentary canal. The complete outer layer is known as the serosal layer. That makes serosal fluid and lubricates the outside, so the organs float around and don't get stuck, all right? So it keeps it lubricated. Then the next layer inside is the muscular layer that we went over in figure 12.22 of the small intestines. Muscular layer has inner and outer muscles. The inner muscles are circular muscles that contract, they go like this. And the outer layer are longitudinal muscles that shorten and push through. Between smushing inward like this and pushing through, that creates a wave of peristalsis that propels food through the intestines. All right, so that's the muscularis layer, muscularis externa. Last but not least, we've got the submucosa and the mucosa. The submucosa is made of like loose connective tissue with little glands and blood vessels throughout it. And then the mucosa is the innermost layer that you see in the figure 12.22, where there's the villi, and it's just a surface epithelial. And it has a little bit of muscle, muscularis mucosa. It also has a lamina propria, these connective layer, but they're all part of a very thin layer of surface epithelial and protective and nourishing tissues. A little connective, a little smooth muscle. That's all the mucosa. So again, what should you know? The outermost layer is serosa, innermost is mucosa, and the muscular layer is what's responsible for peristalsis. And this guy, the submucosa, is just a nourisher. And again, know the definition: wave-like motion of food through the alimentary canal. That's the definition of peristalsis. It's a good word to know. And just to reiterate, I just had my blood test. Triglycerides are digested fats. When you get a blood test, it gives you the good cholesterol, bad cholesterol, HDL, LDL, and your triglycerides. Triglycerides are digested fats, and they combine in figure 12.22. If you look, that's a lacteal, the villus. The villus has little lymph lacteals going into it that absorb fat. And the triglycerides, or digested fats, combine with proteins and are brought to the liver at about 1 liter per minute. And in the liver, the triglycerides are processed into HDLs and LDLs. to travel throughout your blood system. All right. Last but not least, let's talk a little bit about teeth. I have a picture of the teeth in figure 12.18. Probably a better picture than that's in your notes. And I just want you to be aware that your oral health is changing all the time. Oral health is very much based on your hygiene. And I was really happy. My dentist said my teeth are impressive. And they weren't always. As a babe, I don't know what I was doing. I had a bunch of cavities as a little kid. And, I hated the dentist, and almost became a dentist. Very tempting. I like the profession. It's a great profession. I'll leave it at that. But, I had to decide. PhD, become a professor, or go into dentistry. Dentistry is very mechanical. Not that I'm bad with my hands, but, professor, more cerebral, chit-chatting. So, and again, not that you can't talk your patient to sleep as you're drilling them, but ultimately that's my own history. Dentistry versus professor, tough decision. Almost did both, but too many years of school. To make a long story short, let's go over the teeth. The teeth were fascinating to me because they're a continually changing part of your body. And by that, I mean your oral health is very much based on your hygiene. Whoops. What do I mean by that? You're not just born with good teeth. You can be born with the very best structure, and they're going to rot away by the time you're 55 if you don't treat them right. And, smile, be glad you have your teeth. They're expensive to fix, and sometimes they can't be fixed. So let's go over the structure of the teeth. The enamel is the hardest surface we know about. They're made of calcium salts, harder even than bone. Underneath is the bone, the dentin. Below that is the pulp, which is the living blood vessels, nerve, that cavity. And when they give you a root canal, they deaden and clean out that pulp area. That's the root canal. Cementum connects the teeth to the gingiva, or if you remember, the alveolar margins, the bones of your maxilla and mandible. So why discuss teeth as a specialty topic? Because it is something that humans can control. It is linked to other diseases. We talked about the heart. They believe that tooth decay may be linked to damage of the heart, that some of the bacteria damage valves, damage blood vessel walls. So oral health is not just relegated to the mouth. And it's something that needs to continually be addressed. I will tell you, I had an uncle, 96, he was a priest. He never had a cavity. Father Kiden, he was a Capuchin priest, and lived to be 96 and never had a cavity. He would not put toothpaste in his mouth. And he's born 1870, for God's sake, So I just want to tell you that, no, in the 1874, I remember that. But he died when I was just a babe, and lived to be almost 100, let's say. And to make a long story short, he brushed with salt water. He was against fluoride. Fluoride, as you know, is a killer. It is, now you may say, Demfle, you're crazy. Obviously, in small amounts, it's not dangerous. We deduced that over the years. But it took a lot of advertising and research by the American Dental Association to convince people in the 50s and 60s to brush with fluoride, because you're putting a poison in your mouth. And if you just suck down a ton of fluoride, it'll kill you. So my point is, fluoride is in the next class, all right, on Thursday, we're going to talk about fluoride as an electron sucker. It sucks electrons away from bacteria, and that's a way it kills bacteria. And I just want to say, it also strengthens the enamel, but I want to say that is a toxin, much like cyanide, pulls electrons. So there had to be buy-in. People had to change their way of viewing the world. And everyone, I'm going to ask you, how many people brush every morning? You better raise your hand. There's a social stigma if you don't, right? Obviously. And I mean, I brush every morning and every night. I should brush in the middle of the day, but it's been working out pretty good. No cavities for many years. And I just want to say that that's a psychological buy-in to the science, right? We're engaging in something like that now with the virus, although the virus is going to end and tooth decay is here to stay. I can tell you. You know, they put a sealant on with you guys, you're pretty young, so it really is a big protector, but it's no guarantee. And I guess what I'm saying is that buy-in had to happen, and it happened with research and a lot of marketing to get people to brush their teeth. This was not something socially acceptable. 1914, you know, you weren't going to just brush your teeth with fluoride. So, you know, you can look up the history of fluoride if you're bored, but you're not going to do that. But trust me, it's interesting that science is very much not just science and data. You're witnessing this. You're probably living through an era of history that you can tell your grandkids. But I want, you know, if you take anything from the virus and the fluoride discussion, take that science changes society, society changes science. And there has to be buy-in. Like if no one believes that fluoride is going to help their teeth, it ain't going to help people. So that had to be sold. Right now, there's a lot of science and conflict about the virus. So as a result, you know, wear a mask, don't wear a mask. You know, they're very bad for people. They did a terrible job in their communication of what we should do. And, you know, everyone go to the supermarket, don't wear a mask, don't go anywhere else, funnel in. And again, it's not a virus conversation. This is a digestion conversation. But, you know, I like to draw parallels with you to see you're living in history, and science is at war with itself. Fascinating. And there was a time, again, with tooth decay and fluoride where this was the case. Obviously not as serious. You're not running around dying of tooth decay. But nonetheless, something to think about. All right, moving along, last but not least, in the interest of time, salivary glands. I just want to address that in the mouth, there is 3 types of salivary glands. The parotid, which is large and back here, and it makes your amylase. The sublingual, which is under your tongue, makes mucus. And the submandibular, which is smaller than the parotid and under the mandibular area, makes amylase and mucus. All right, each of them make a different type, a different amount of each of these, mucus or amylase. And it happens to be that serous cells of these salivary glands produce amylase, and the mucus cells make, get this wrong on the test, you got problems, mucus. How do we control the rate at which we digest? 3 hormones that you should be aware of. And my favorite is CCK, so I'm going to start with that. All right. These hormones regulate how quickly you digest food. And it's the rate of your digestion is based on what foods come into your body and how your hormones react. So when fat enters the small intestines, in other words, where there's an increase in bile releasing into the small intestines due to fats, intestinal walls, in response to those fats in the duodenum, will produce CCK, cholecystokinin. So in other words, fat in the small intestine stimulates CCK to be produced. And that causes peristalsis to slow and bile to increase in its release from the gallbladder to the small intestines. So a slower rate. Now why? Because it's much easier to digest a carrot, you know, a bunch of carbs, than it is to digest those long fat molecules, all right? So CCK slows down, in summary, CCK slows down your intestines, peristalsis, and your stomach peristalsis when fat enters the duodenum, all right? And bile increases in its release. Next step, secretin. Secretin, when an acidic pH of chyme is just the mucousy ball of food coming from the stomach. And when that chyme and its acidity goes from stomach to small intestines, the intestinal walls make secretin. And secretin causes the pancreas to release bicarbonate. And that neutralizes that acidic chyme or bolus of food. Another word for a round ball of food is a bolus. Enterogastrone slows the movement of food into the small intestines in fat's presence as well. So CCK and enterogastrone act in tandem together to slow movement of food. All right. I suppose we could say CCK causes the stomach to slow and enterogastrone causes the small intestine to slow. But what should you know? You should know that both of them just slow intestinal and stomach digestion in the presence of fat. And that is our hormonal control of food. And that concludes the lecture. Let me open it up to discussion at